Welcome everyone to our seminar series, Molecular Communication Outside the Box, Breaking Disciplinary Boundaries, organized by the IEEE Technical Committee on NBMC. I'm Max, you all might know me, and I'm the host for today, at least for the start, and then later Werner will host the questions. We are very happy today to welcome Martin Frauenlob from the Technical University in Vienna. So this is also in Austria. And maybe before we start, a little bit about Martin. So I also get to know him just a few minutes ago. So he got his PhD in 2019 at the Hokkaido University, and now he is a research group leader within the cell ship research group in Vienna University, with a research focus on biomedical engineering and microfluidics. And he's working a lot in 3D cell cultures, biomaterial synthesis, and microphysiological systems. So I'm sure that this will be very interesting for the community for possible collaborations, or at least get to know a little bit about the research from people from lab practice. So Martin, the stage is yours, and we are looking forward to your talk.
Okay, thank you very much for the introduction. Yeah, as you mentioned, today we'll talk a bit about organ-on-chip technology. I guess some of you, a lot of people know this keyword, but what is meant by it is sometimes a little bit unclear. And today the topic should be a bit how we can utilize this equipment in toxicology and pharmacology. And for that, I created this talk. It's a bit of a lecture-like talk. So we'll talk about what is toxicology and what the key principles and where then our research group comes into play, for example, or where there's nowadays still limitations and how we can overcome that. So, and if you have questions, just feel free to just interrupt me. This is no problem. Yeah. So to start, we are talking today about toxicology and pharmacology. So the meaning is, okay, what is toxicology? It's really the science of understanding how chemicals affect the human body. So that's often about those responsive relationship of compounds, and same goes for pharmacology. So you have a focus here on drugs and their therapeutic effect, and there often it's mostly important to go through these four key domains: the absorption, distribution, the metabolism, and the excretion of these compounds. And this is often combined under the keyword ADME. And what these two disciplines share, toxicological and pharmacological studies, is that both of them are highly dependent on those those dependent effects. And the second thing is both of these fields are mostly concerned with the safety of organisms, and most of them are also about the communication between organs or the transport of compounds between organs. So here we need to talk a little bit about organ crosstalk. So what is meant by that? Organ crosstalk is basically the dynamic bidirectional communication between different organs or tissues in the human body. And this communication is done obviously by different types of molecules, for example, hormones, cytokines, and various metabolites, and then also the neuronal signaling. So on the right side here, you just see an illustration of absorption organs we have in the body, for example, the skin, the intestine, and the lung. What I want to emphasize here with this figure is that all of these interfaces look highly different. And when we have to think about organ crosstalk, is that these are all different entities, and all of them have different structures and functions. And however, there's always feedback loops between these organs, to how they control each other. And a lot of these things are not properly understood because they're extremely complex and we cannot look properly into a human body. So when we have a dysregulation of certain crosstalks, this can contribute or cause diseases. And through drugging, we try to cure these diseases. However, there's always this issue that when we give a patient certain drug, there is unintended actions that happen in the body because it's still not fully understood. And here, I just would like to give one brief example because one of my students encountered it lately in the laboratory. So he's working on a microfluidic device where he's trying to use different compounds in an organ chip system. And here he wants to, for example, put paracetamol into the culture medium and investigate how it affects multiple organs. And then he realized that if he's adding paracetamol to his system, that blood-brain barrier cells, as you can see here, at very low concentration of paracetamol immediately start to die. And here, I think it's important to talk about these on-target, off-target effects and what it's meant by that. So we know that diseases function a certain way, and you create a drug that either blocks a certain metabolite to be created or enforce a certain metabolite to be created more. However, this molecule that you're adding to the body can also function in a totally different way in another part, in another organ. For example, here, in the case of paracetamol, we know that in the liver, paracetamol is enzymatically digested by cytochrome P450 into NAPQI, and this compound is quite toxic for the cell itself. However, in the liver, we have a lot of glutathione, and with glutathione, this reacts to cysteine and mercapturic acid. So this is not a problem for liver cells because they actually produce a lot of glutathione. However, if paracetamol ends up in the bloodstream, then it can get in contact with blood-brain barrier cells. The good thing is, the bad thing is, the blood-brain barrier cells also have this protein which creates the NAPQ. However, they do not produce glutathione, and in the end, paracetamol, if it would at a high concentration go into the bloodstream, then it would actually have a toxic effect on cells that are in the interface between your brain and the bloodstream. And so in many ways, drugs can be toxic for the human body. And that's why toxicology and pharmacology always go hand in hand. And so here it's always really important that we investigate where these compounds end up and how long they stay there. So that's where pharmacokinetics come into play, because there you actually want to analyze the peak concentration of a compound and how long it takes this compound to get to the location where it's needed, and how long it takes that this compound is actually excreted from the human body. And here you can see an overview based on different types of oral addition of a compound, and sometimes it depends if you add a tablet or a capsule, obviously, because the capsule is, no, it should be, yeah. I will not talk about this in detail, but overall, what we have a system where a drug comes into your body, often through the intestine, then it penetrates the epithelial layer, then here something is metabolized, then it actually gets into the bloodstream, into the liver, there's also something metabolized, and then it gets actually maybe to the place where it's needed, and that's where it should do its action, and that's the most important part, because only if we know how many, what's the concentration of the bioavailable dose, then we can find out, or then we can be sure that this amount of drug can be metabolized at the point of need. So when you look at pharmacokinetics, you can model that. So how can you model that with, so there's the physiologically based pharmacokinetics. It's like a predictive model for this absorption, distribution, metabolism, and excretion. And here you have basically different parameters, for example, the blood flow and the absorption and where absorption happens. So at first, for example, you have in the stomach, then it goes into the intestine. In the intestine, the drug is absorbed through the epithelial layer, and then a certain volume stream of blood goes into different compartments of the body, and there it's also metabolized. However, this is very nice to have because that's how we create the model, but for that you need key parameters to calculate it. And these key parameters, the question is, where do we find them, or how can we get to them? And this is still done with animal models. So, I mean, animal models are the gold standard for drug testing, but they have still major drawbacks. Like, for example, here we have an example, the reproducibility of the effect in humans, because in the human body, proteins often work differently compared to animal models. And for example, here we have the YOX medicine. It was brought to the market as an anti-inflammatory drug, and it was rejected in 2004 because it was found that it has a severe risk of heart attack, and it was estimated that 27,000 people died in the period where it was actually sold. The other part why animal models are not the best choice is still, or is now more and more emerging, because it's an ethical issue. Why should animals actually die for human cause? So overall, there's a lot of animal testing going on. So in the in Europe, we have 9 million animal tests per year. Most of them are for fundamental research and pharmaceutical research as well. And it is always one of the major bottlenecks of getting a drug to the market, because at first you have the clinical studies, before you have the clinical studies, you have the animal testing, and then you have the clinical studies. And the good thing is, up until the animal model, everything is working fine. And when you go to clinical studies, often complications happen. And for that, here we have just some numbers. For example, often the data that you get from animal models is misleading, because you have just around 50% of mouse tests are actually reproducible in the human, and some of them are even acute toxic for the human body. So that's why 9 out of 10 drug candidates actually fail in the clinical trial, and before they actually looked good in the animal testing study. And one agency who is now trying to get rid of, or who is pushing towards getting rid of animal models is the 3S movement, which basically means reuse, recycle, and reduce. And they pushed for many years to get rid of animal models. And then in 2022, one of these major goals was achieved, on this paradigm shift, that the FDA, so the Food and Drug Administration in the US, is accepting new alternative methods for non-clinical tests in preclinical studies. And there, one of the biggest things was that you can now actually use organ on chips, on so-called microphysiological systems, to actually do testing or do drug testing on. which is a huge boost for this whole field. And when we look at this field, like for example, do a Scopus search on organ-on-chip systems, then we see that after 2013, like this field really exploded, and now we are where we are. But the question is, how did we get there? Or how did this field actually evolve, organ-on-chip technology systems? So it comes like every technological improvement. It goes through many steps, and often the steps include miniaturization. Because when you think of organ-on-chip systems, this is a laboratory and you just shrink it down. For IT, information technology, you saw the same trend based off the same reason. It's often economic reasons and market demand. And what you want to have is that technology is portable and energy efficient. And this can mostly be achieved by miniaturizing and integrating and automating things. I think one of the best examples was, well, general industry, industry in general, but then information technology. Another thing is analytical technology. When we look at how UV-Vis spectrometer developed over time, from the 60s up to now, they decreased highly in size. And for animal studies, the question is, okay, how can we reduce that? And there, before the organ-on-chip committee was founded, there was the lab-on-chip community. So the idea would be, or the question would be, okay, who needs actually a laboratory? Because in a laboratory you have to work. Wouldn't it be just nicer that everything is automated and you actually do not need a laboratory? So that's why they were thinking on how can we integrate processes that people are actually doing in the laboratory, miniaturize them, automate them. And this was done by, sorry. And this can be easily done. And there's a lot of researchers working on different ways of mixing, of heat transfer, of storage units, or like how to develop all of these things that we can automate all the laboratory things that all the things that are done in the laboratory, how you can miniaturize that and put them on a microfluidic device. And then out of that, the organ-on-chip field was developed. Maybe somebody knows this researcher, or maybe not. It's Donald Ingber. I think he was the one who actually started to talk about organ-on-chip. And I think he defined it as the combination of lab-on-chip technology. So when we have the fabrication of these microfluidic units, then the implementation of sensing units, and then the final thing is adding the biological tissue. And this combination is then an organ-on-chip. Why are you doing that? Because in a microfluidic device, you have a small volume. So often an issue is with cell culture or biological tissue, you have to add a lot of very expensive compounds to these cells so that they can survive. So if you reduce that, it's just much easier for you to get the laboratory running on a low budget. Another thing is that you can recreate much better a three-dimensional multicellular architecture in these systems. And this helps very much when you look at how diseases, how a disease is located in the body. It's not mostly just one tiny tissue that is inflamed or so. It goes through many tissue layers. And these layers you actually can place next to each other in an organ-on-chip device. And then you can just reproduce certain diseases much better on an organ-on-chip device. So talking about this, I think one thing that needs to be noted, the organ-on-chip field is quite an interdisciplinary field where you have engineers and fabrication engineers, you have the biologists, and you have polymer scientists, and in between and the overlapping part is the multidisciplinary field of organ on a chip. And me myself, I come more from the biological part and from the polymer part, and I'm not too familiar with the fabrication and the sensing itself. And I think the hardest part in this interdisciplinary field is that you actually can create a research group where you have all the knowledge, where you have actually employees and partners who are willing to work on these fields and not on their main research topic anymore. So how can we use the organ chip system for drug testing and toxicology studies, or where should we place it? When you look at such studies, you have in vitro studies, so very simple models where you just put human cells on a plastic dish and add the drug and see how they react. And on the other hand, you have the very complex in vivo models that are not very close to humans, but they represent the complexity of a human body. And a microfluidic system is basically the merger of these two, or it should get the bridge between these two worlds. Because in a microfluidic system, you can actually, through this multicellular architecture, or like having multiple channels next to each other, you can build up a certain complexity. You can also have an inlet flow, outlet flow. You can measure parameters much better. So what can we monitor? What is better in an organ chip device? One thing is you can introduce mechanical forces into systems. One thing is, okay, we know that the blood vessel wall is built out of endothelial cells, and these cells actually need to be exposed to a certain shear stress so that they actually express a phenotype that is found in the human body. Then one other thing that we can very well show is that these transport phenomena when we have diffusional mixing. So through diffusional mixing, we can create very nice gradients in our systems, and then we can very easily show, for example, when you have here the system, cells on the left side are much more exposed to a certain compound than cells on the right side than on the left side. And then if these cells would die, then obviously we'll see that there's a concentration-based effect. That's something that we can do very nicely. And then another thing is integrating this architecture that I talked about already. So we have like these biointerfaces where we have different tissues next to each other. And when we look at how cell culture was previously done, so you have just a material here on the bottom, mostly it's polystyrene. It has a super high stiffness, and you culture cells on it. You use immortalized cell lines here. So this system is really extremely far away of the human body. And if you go into this organ chip systems, you have cells that are in 3D encapsulated in a 3D matrix. In this matrix you can tune, and because of this progress in polymer science, you can also use primary cells and so on. So with organ chips, you can just be much closer to the human system. Here's just a short overview of what can be used, or what is used to recreate the in vivo situation. So one thing is the 3D environment that we can modify. That is mostly done with various types of hydrogels based on either synthetic materials like polyethylene glycol or PMMA. PMMA not, because this is a stiff polymer. but polylactic acids too is a hydrogel. And then we have the natural polymers like collagen and fibrin and so on. Then with these materials, some of them are degradable by human cells. And when you read publications, you have to be a little bit, you should know that, for example, when people talk about collagen, scaffolds or collagen hydrogel, this can mean many things because it depends on how you cast it, if it's actually hydrogel or if it's a sponge. If it's a sponge, it has much different properties compared to hydrogel. So sometimes this is a bit unclear and we have to take care of that. Then another thing that you can emulate is the cellular heterogeneity, because we can very well control the culture media composition in these devices. You can actually choose primary cell lines to actually recreate the systems that we see here. So we have primary cells, we have stem cells, and with the combination of these primary cells and stem cells, we actually can recreate this kind of niches in between tissues, which are very important for this organ crosstalk because there a lot of processes are happening. Another thing to consider is also the oxygen concentration that you found in the human body, because in the human body, the oxygen concentration is changing drastically between organs, from the lung, obviously, where we have the highest concentration, down to a very low concentration or extremely low concentration in tumors, because there the tissue is so dense that diffusion of oxygen is very hard there. Then the last thing is also what you can mimic is various mechanical stimuli that come from mechanical properties that are introduced by topology, curvature, and also mechanical deformation that is very much needed for muscle tissue. Especially, I think I was lately reading a publication where they were actually showing in a microfluidic device that it's extremely important that after injuring a muscle, the muscle still needs to be moved, or else the regeneration cascade is not starting properly. So this, in conclusion, means that if we go back to the pharmacokinetic models, we actually try with oil chip devices to replace animal testing. And for that, a lot of research is done on particular organs, and some of the research is actually done on whole body on a chip systems. So what is that? You basically have a microfluidic device where you have a circulating system, and then you have compartments for each kind of cell type. And then you introduce the drug into the bloodstream, and then you investigate where it's localized afterwards. And one of the bigger publications that was done or published lately is not maybe the exact term, but 2022. But I think this publication has gotten a lot of attention, is this multi-organ on a chip system for pharmacokinetics. So here they introduced a system. Everything was here 3D printed with the upper part, where you have compartments for all the different cell lines. They were all using primary cells here, and all of these primary cells need different conditions. As you can see here, for skin, it's actually an air-liquid interface. For heart, it's actually a muscle that is actuated, so it behaves in vivo-like. And for bone, it's a very stiff 3D matrix, and for liver, it's a soft 3D matrix. And they culture all these cells in these things, in these small wells, and then they have a bloodstream that goes below. And this stream was just connected to a peristaltic pump, and with that they they actually can create a certain shear stress on the bottom of of each compartment. So the shear stress is important that the endothelial cells that are cultured there behave in vivo like, so meaning that they have these elongated characteristics. And then the nice thing is that they actually can, for example, distribute um, have multiple pumps here in the system and have a blood flow between all of these compartments over and over again. The idea is that at a certain point they take out one chip and bring it to the analysis so that they have multiple time points. And one thing, and this is the whole setup, this is the peristaltic part. And for people who work in cell culture, I think this is very shocking because this is a extremely old incubator, and this is part of the publication they have shown. And what I want to emphasize here is that so these organ on chip systems, you do not need to have a very clean laboratory. And I think that's a good thing because the more dirty you can work, the more easy it is to utilize these things somewhere else. So I love this figure. This is a supplementary figure where they actually show that these experiments were done in extremely dirty conditions. So the next thing is that they can analyze an extreme amount of things. So they have here immunofluorescent staining data where they can show that their heart is behaving like a heart and so on, the skin is behaving like the skin. Then they have proteomics data that show very similar things and that they have for two different time points, for time point 0 and time point 24 days. Then they have experimental data that is where they compare the experimental data with the model, and they can actually show. So here they used the chemotherapeutic drug doxorubicin, so they added it into the circulation and checked then the concentration of the doxorubicin in their system over time, and they just showed very well. This fits nicely to our model, and they have also here 3 different time points. And I think that's where our research group comes into play because when you go back, okay, like you have experimental data, but just from the beginning and the end, then here you have official 3 data points. It's not much. And I feel like that's where our research group comes into play because when we look again at this, what is still missing is actually that we look more into time resolved data sets. So the problem with these biological analytical tools, most of them are endpoint analysis things. So what you do, you take all the biology out of your system and then measure gene expression. And if you do that, you destroy everything. And our philosophy would be that we measure, so we integrate sensors properly into our system. So this is the setup of our research group. We have on one hand the rapid prototyping team that focuses mainly on the fabrication part, then the lab on chip integration team, which is focusing on the introduction of sensing systems. Then we have one smaller group that is focused on diagnostic and point of care devices. And then we have 2 different groups that one is focusing on disease models, on these organ-on-chip technology systems, and the other group focuses on the microtox studies. And here I would like to talk a bit more about the electrical sensing, because I feel like that's one of the most important parts that is often not properly done. And that's why organ-on-chip technology is still not cannot be properly used in industry right now, because a lot of sensing is not properly developed. So why are we doing sensing? So one thing is to probe the microenvironment around the biological tissue, but also probe the biological tissue itself through electrical sensing, electrochemical sensing, or optical sensing. And in our research group, we focus a lot on the implementation of the impedance-based measurements. So what is that? So impedance is basically the electrical resistance in alternating current, and this resistance. So if in this electrical flow field the resistance of the medium is changing, that can be measured. So for the impedance, you always have the real part and the imaginary part. And at high frequencies, you actually introduce this, you polarize particles and they deform the electromagnetic fields. And through that, you actually can measure many different things. For example, here we have an electrode on the bottom. We have the sensing electrode, so negative terminal and positive terminal, and on top we are capturing cells, and they are actually deforming the electromagnetic field and change the resistance, and then we actually can measure how many cells we have here or how many cells we do not have there. So the idea would be that we can get rid of all the optical sensing in our system, because the optical sensing takes up a lot of space. Not everybody can afford to have a huge fancy microscope in the laboratory. So what we try to do here is integrating these things and then investigate, can we measure growth phase? Can we treat the cells, and what is happening afterwards? Then the other sensors we're focusing on are multi-electrode arrays, multi-electrode potentiometry. So here, the idea would be that we have mainly neuron cells, and these neuron cells are cultured on these things, and if they start firing, we can measure electrophysiological signal. So I think I should skip these things a bit, because one limitation we often reach is that electrodes are mostly printed on glass. And when we think of biological systems, a glass, or glass is not an interface, because when we want to investigate how cells or how a barrier is changing, we have cells on the top and cells on the bottom, and this is not possible with glass, obviously. So the idea would be that we actually need to integrate sensors into a porous membrane. And how can we do that? Here is an example. So here we have a so-called placenta on a chip, and the idea would be we want to investigate the nanotoxicity of certain molecules on this barrier. And for that, our team developed a go-to-position protocol for porous PET membranes, and they're sort of, after they developed the protocol, okay, the electrodes work, then they just did cyclic volt thermogram, and then show, okay, we don't have any dissipation. Then based on the width of the electrodes, we can probe a different the electromagnetic field in different depth, and then we can culture cells on it. And on the right side here, we can measure, for example, how the diffusion through this membrane changes if we do not have cells or if we have cells. And what you can see is that if we have cells, the diffusion of different sized molecules is very slow. Then here's just the data set for the sensors. The electrodes with distance between the positive and negative of 50 micrometers and 100 micrometers. And there you can just see if we add cells, with the 50 micrometers, we can not measure much at the lowest concentration, but with the 100 micrometers, we can, because the electromagnetic field penetrates deeper into or further away from the electrodes up to around 100 micrometers. And then the final study was in the end, okay, we have our system, we have cells on it, and now we're adding these molecules that induce nanotoxicological stress. And there we can see if we add it at a certain concentration, the impedance signal drops because the cells are dying, and for the control and low concentrations, the signal stays kind of constant. And this was confirmed with a cell viability study. So this shows that our chip is actually functional, and then we can play around with that. So we can add different types of cells on the top, different types of co-culture or on the bottom. And what is nicely observable that we actually have a much better time resolution, because here we can measure the same chip on different days, so we get much more data points, and we get the information how dense this tissue is or how weak it gets. And now going shortly into the biological part, or what is one of the bigger topics in our research group, is this gut-brain axis. So why is the gut-brain axis important? I think everybody knows now that there's or it is assumed that the microbiome from the gut actually has an impact on certain neurological diseases, but also some mental health issues such as depression. And for that, we actually need to recreate the brain and the gut. And so here you have the interface of the gut. So the first thing, you have the epithelial cells, and below you have the enteric nervous system cells, which in the end transmit the signal up to the brain. And for neurodegenerative diseases like Parkinson and Alzheimer's, we often have patients that are old and not young. So what a student now is working on, a microfluidics system with a sensor integrated membrane, where you can actually add human cells on the electrodes, and then, I mean, she showed here that, well, we can do that, everything is fine. And then the question is, how can we make these cells older so that these cells behave like an old person in the end? So the idea would be to add again a cytostatic agents, in this case doxorubicin. So there she was just investigating how different doxorubicin concentrations affecting the senescence and viability of cells. So for example, here if she adds more doxorubicin, they start to express senescence markers, but actually they're still kind of okay, so it's still living. And then putting this compound on the microfluidic device, she was able to show that actually the signal is changing. So that, for example, in the case if she has doxorubicin, the impedance signal is increasing, which means that the membrane is less permeable, meaning maybe this means that the cell membrane might be less and less flexible, and through that, this can happen that the signals increased. And when we look at the PCR data, she found out that certain markers are upregulated, and these markers are related to an aging intestinal cell. But these markers are very similar to biopsy samples that you can get from older patients. And then the second part here is the Parkinson's disease model that we are working on. So Parkinson's disease is a very common neurodegenerative disease that is found in our elderly society almost in every family. So the thing is that they have all these like tremor symptoms and depression and even hallucinations at a certain point. And how, what do we know about this disease? We have this loss of these dopaminergic neurons, and we have this accumulation of the alpha-synuclein. This is a protein in the midbrain, and then the neurological signaling is it's much slower. And for that, we have created a microfluidic device. So we basically, so maybe just show this device. We have here 3 chambers: left chamber, middle chamber, right chamber. In the middle chamber, we put basically a mini brain there. So these mini brains are actually based on stem cells that we get from patients. We differentiate these stem cells into brain organoids. It's basically a clump of cells that shows a brain phenotype. And we put them on our microfluidic system. And the nice thing is, in the microfluidic system, we can assert a certain flow rate from left to right. And this here is quite important because this mimics this interstitial flow of liquid in the brain. And this flow is in nighttime quite fast, and during the daytime it's quite slow. And that's what we also can mimic in our system. And if we do not that, the cells tend to die. So that's the comparison here. So here's the dynamic system where we have we shake our chip and we create an interstitial flow. And if we do not shake the chip, then we get a much higher signal here. The Cas3 signal of the static culture is much higher, meaning a lot of depth in the system, and in the dynamic it's not. And it also has an impact on the neuronal outgrowth in this system. So we have, in the static condition, we always have, like this is the neuronal outgrowth here, and this outgrowth at first follows the interstitial flow, and it also shows a more in vivo-like phenotype. And it's also important because this neuronal outgrowth is actually connecting to the electrodes, and then we can actually properly measure something. So then just some more slides and then I will be finished. So what we have here is a sensing tool based on optical sensors. Here we actually can measure O2 concentration or also pH in our system, and we know that an active brain actually consumes a lot of oxygen. The oxygen demand is very high, and there we were just trying to analyze, okay, how is the oxygen demand changing over time? And since the cells are growing, we assume that more oxygen is consumed, and that's what we were able to show. Up to 28 days, the oxygen demand was increasing. And what is also known, that diseased, so people with Parkinson have or brains with Parkinson, they have a lower oxygen consumption rate, and that's what we tried to compare. So we put diseased midbrains on our device and healthy midbrains on our device and compared the oxygen consumption rate. And there we were able to show that the wild type has a much higher oxygen demand than the disease model. Here I just have some more pictures how it looks like. So like this is a chip where we have the electrodes on the left and right side and the neurons that grow towards these electrodes, and then we can measure this electrophysiological signal. Here you can see this is the edge of the organoid, and these are the neurons that grow towards the electrodes, and these are the potentiometric signals that we can get from the firing. And the idea would be that also the firing in the Parkinson patients is weaker or slower, but that's something that we are still working on. And so now I have shown the gut system the brain system, both of them have integrated sensors. And the idea would be now to combine these two systems. That's another project that student is working on. So we have here two circulation systems with micro pumps here, left and right. This is the circulation for the brain. The other one is the circulation for the gut system. And then we have basically a pump that we can control. Then we have sensing to investigate what's happening to the gut and to the brain. And now we can just add a drug and live monitor what is going on in this whole system over an extended period of time. And I think that's where pharmacology and toxicology should head for, because this might give much more interesting experimental data. And that's what we are doing right now. And yeah, this is all my research group is working on. This is the building we are in, and this is the research group. So the gut work is done by Constanze here, and a lot of brain work is done here by Alexandra and also by Sophie. And Matteo is doing the combination of both systems. And yeah, that's all from my side. And I hope there was something that was interesting to you in this talk. Thank you very much.
Okay, thanks, Martin, for a very interesting talk. I think there are a couple of questions from participants. Jorge, please.
Yes, thank you. Thank you very much, Martin, and very nice talk, an amazing technology and progress there. I'm wondering if there is any limitation due to the scale in size, in the way that, for instance, if we want to talk about the impact of one organ with another organ, this communication among them, in the organ on the chip test, that these two organs are quite close placed, let's say in the millimeter scale. However, in the human body, they might be in the centimeter scale. They might be by far away, one organ from the other. Then, is there a way to compensate for this, or there is no way, and we simply talk about local happening in a single organ, and we're not talking about what is happening in this long connection in the human body?
I mean, that's a very valid question. Overall, you have to assume obviously everything's working much faster, and also size plays a very important role, because there's always the question, when is a tissue functional? Or when does a single cell, so you have one single cell, it comes from the liver, when does this thing, how many cells in addition do you need that it behaves like tissue? So it's often, when does the function appear of this tissue? And that's honestly still an ongoing question. And there's many ways to answer it. You can say, okay, like we take gene expressions from the liver that we find in biopsies, and compare our systems to at what point do these phenotypes match. But the gene expression phenotype is an RNA signaling. So this does not always translate to what kind of proteins are present. So then you would need to go to proteomics maybe. That plays a role. So yes, it definitely plays a role, the size of the system, because you have different diffusion and so on. And for the distance, I would say you can compensate for it based on modifying the fluid flow.
I see. And regarding the volume, I can imagine there is a second limitation, because if we consider realistic system where biocomponents degrades, it's not the same when these biocomponents are flowing in a bigger volume, where this degradation rate might happen faster than in this test bed where the volume is little and then there is little degradation rate in the system.
Yeah, definitely. There's also that I think the biggest issue what you have with organ chip systems is the inlet and the outlet. How do you define what should go in and at what point? So because in the end you have a circular system. This is your organ chip system. But what you now do is often that at a certain point you just add culture medium and take something out. This is not what happens in the human body, because only if a certain concentration of something is reached, then it passes through the liver membrane and then, or the kidney membrane, and then it's excreted from the body. And this we need to look at much more closely. When to get things in and when to get things out. I think this is really good.
Thank you.
Okay, are there any other questions?
I have a question. On slide 51, I think.
51.
It was about a mini brain which was behaving differently at day or at night. In the previous one, yes. Could you explain what exactly was observed and then why?
So maybe I wasn't totally clear. You have here in the human body, you have a certain blood pressure, and because of the certain blood pressure, you have fluid flow within tissue. So and you also have the lymphatic system, which is opening up during the night process and shutting down during day. So like I think that there's this when you didn't have a good night's sleep, you have a headache. This comes from the fact that the lymphatic system wasn't working properly because it was closing, so it was preventing the flow through the tissue. And what we can show here is actually that, or what we try to do here is that in a normal human body, when you sleep, you have much more interstitial flow, and during the day you have less interstitial flow. And the idea would just be here that, okay, with our tilting mechanism we have, because we have a microfluidic device, we put it at a certain angle, so we have hydrostatic pressure, and then we let it flow. Then we have just a really high flow rate through this tissue, which also means that a lot of nutrients are coming to the tissue. And when you have a certain clump of cells, and a limiting factor is diffusion, and around this clump of cells you have a shell. There is also the issue that it's based on diffusion, so you always want to get as much fresh culture medium as close to the cell bulk, so that you actually have a proper nutrient supply. And I mean, that's why we're doing this, because in the end, if you have a certain bunch of cells, and if you do not add enough culture medium or get culture medium close, they die, what was shown in the next figure. So this like day and night cycle is kind of this periodical nutrient supply that you need for a system.
So during night, the body has enough resources to open up and to let the nutrients flow. And during the day, it's not possible.
Yeah, it's slower.
It's lower. Okay. Yeah. Thank you.
Okay, any other questions? Maybe a last one from my side. Do you have an idea if there's any ongoing work using such an organ chip platform for targeted drug delivery?
Targeted drug delivery? Yes, I think that there is stuff going on, not in our laboratory, but what is often also happening, or what was a nice thing to do, they took drugs that failed in the clinical trial too, where they're using people already, and where they saw, for example, in this person, there was like, as mentioned, for this one drug, it was the risk for heart attack. Why does the risk of heart attack appear? Normally because, I don't know, platelets are attaching to the vessel, and then they're clogging the. And with microfluidic devices, they were always trying to show that look, I have a microfluidic system, and if I add this drug, the same thing is happening as it happened to this patient. Like these things are done quite frequently now. And I mean, yeah.
Okay, thanks. Are there any more questions? If this is not the case, Martin, thanks again for giving this very interesting talk. Yeah, that's the end of the webinar. Have a nice day and hope to see you all in the next webinar. Thanks and goodbye.
Bye.